So in this video, I'm going to go over the science shown in episode 3 of Netflix's BIOHACKERS TV show. If you haven't seen my videos reviewing the science of the first and second episodes, definitely check those out as well. But for now, let's just jump into it. First up, we have the GFP Weed scene, where Mia, Jasper, and the roommates all go to a party with GFP-modified weed plants. This example is even worse than the GFP mouse-head plant from the first episode. Even if we ignore the fluorescence versus bioluminescence issue, there is no way that the GFP just rubs off the plants like some blacklight paint. It looks cool, but that's just ridiculous. And I know it's necessary for the plot of the episode, but I think they could have found a better way to make the plot work. Second, we have the modified mosquito scene, where Jasper shows Mia the mosquitoes he modified to transmit diseases. I really have no idea why you would want to modify mosquitoes to transmit diseases as efficiently as possible. If you wanted to test diseases, you would do it as targeted as possible, and just use the virus or bacteria directly. Adding mosquitoes is unnecessarily complicated, and you just end up with a highly dangerous product with no added benefits. I'm not saying it's not possible, just that it doesn't really make any sense in this context. Lastly, we have the saving Jasper scene, where Mia tries to save Jasper after he gets sick from testing a gene therapy on himself. You don't just inject RNA or DNA for gene therapy. They would get broken down rapidly in that environment, and even if they weren't broken down, the RNA and DNA wouldn't naturally pass the lipid bilayer and enter cells because of their negative charge. With viral gene therapy, the RNA or DNA is encapsulated with viral proteins. These would likely be what's causing the immune reaction and not the RNA or DNA itself. The immune system has spent a long time adapting to keep viruses out, and that's what a gene vector looks like to it. Jasper could have also used lipid nanoparticles to encapsulate the RNA or DNA. The lipids making up the nanoparticles are theoretically less immunogenic than the viral particles, but they're often less efficient at transfecting cells and can be toxic to the cells. If enough cells die, you could potentially get a similar reaction from the immune system. So I guess the lipid particles could also have caused Jasper's fever if he was using this method. In these cases, you probably wouldn't want to try creating an antisense RNA to fix the issue. An immune reaction like this, and what seems potentially like a cytokine storm, could be addressed directly. Some type of immunosuppressive could be used, like tuxelimab, for example. I'm not a doctor though, so I can't really diagnose the patient. I'm not really going to go into too much detail about the lab work they're showing, since it really doesn't make much sense for the situation. Initially, they're talking about RNA and synthesizing an antisense strand to neutralize it, but then Mia goes and performs what looks like PCR. You really don't amplify RNA with PCR. PCR is almost exclusively for DNA. You could do reverse transcription PCR to turn RNA into DNA and amplify it, but that wouldn't allow you to synthesize single-stranded RNA. Also, the PCR they show goes super fast. Usually PCR takes hours, so I'm not even sure they're doing PCR. Then they skip over the gel electrophoresis pretty quickly and go straight to the gel imaging. The benchmark gel supposedly has multiple bands and multiple lanes, but presumably we only want a single antisense strand. Again, I don't really know what's meant to be shown here. Then when the experiment doesn't work for Mia and she calls in Chen-Lu to help, they start talking about CRISPR. Now I really have absolutely no idea where things are going at this point, since CRISPR is completely unrelated to anything we've seen up to this point. So at this point, I just gave up on trying to make sense of what was going on. In conclusion, I think the scientific errors in this episode were maybe the worst so far in the series. The GFP weed scenes were even worse than in the first episode, where they just confused fluorescence with bioluminescence. In this episode, they made it seem that just touching the GFP plants would essentially give you a bioluminescent body paint. It was clearly just necessary for the plot. Similarly, I think the modified mosquitoes were also more a plot device than anything else. Then in the saving Jasper scene, I was never able to figure out how any of what they were supposedly doing was connected to the antidote for Jasper. Basically, this episode was a bit of a mess in terms of the science. But if you liked these videos reviewing it, stay tuned for the review of episode 4 coming soon.